Hi everyone, welcome to Biology 270, Microbiology. This is our first lecture, Lecture 1, Overview of Microbiology. And so this is going to kind of come from two chapters from our textbook from OpenStax, Microbiology. So Chapter 1, and then Chapter 2, but only sections 2.3 to 2.4. So I'll, talk about that when we get to that section. So let's start with an overview of microbiology. And so for each lecture, there are going to be learning objectives like this. And so these are basically the goals of what you can actually do and hopefully know with the information provided. And so you can think of the learning objectives a little bit like a study guide. Like for each lecture, this is like the study guide points. Like you could almost treat these as practice questions. Try to describe how microorganisms are classified and distinguished as unique species. List the various types of microorganisms and describe their defining characteristics. Give examples of different types of cellular and viral microorganisms and infectious agents. Describe the similarities and differences between archaea and bacteria. Describe key historical events associated with the birth of microbiology. Provide an overview of the field of microbiology, and describe the distinguishing features and typical uses for different types of microscopes. So these are all the things we're going to cover. And at the end, you can kind of come back and review, oh, okay, now I feel like I can do this, and let me practice doing that. So to start, what is a microbe or a microorganism? So microorganisms, we also refer to as microbes for short, are generally organisms that are too small to be seen without a microscope. You need a microscope to be able to actually observe them. Whether or not you can see them, they still exist and they're alive. So we kind of categorize these, with this term, a microorganism, that you need a microscope to be able to see and study. And so these include bacteria, of course, but also fungi, algae, protozoans, and viruses. And so I'm going to talk more about all of those a little bit later in this lecture, but those are all categorized as microbes because you need basically a microscope to be able to see and observe them. So humans have been living with and using microorganisms for a lot longer than we've been able to see them. So before we even saw them and knew that they were there, humans have had some idea that there is some process going on, and maybe they didn't know it was due to a microorganism or an organism, but there's some process going on that people have been using for a long, long time. So people across the world have enjoyed fermented foods and drinks for all of recorded history. And so fermentation is a process that requires a microbe, a microorganism to do that process and have that outcome. So making these kinds of foods that are fermented requires microbial fermentation. This process that uses either bacteria, mold, or yeast to convert sugars, a form of carbohydrate, into other kinds of molecules like alcohols, gases, and organic acids. People use fermentation for different purposes, and so you might use different organisms, but there has to be some organism doing that process of fermentation. And so we'll talk more about fermentation when we talk about metabolism. So microbes have been around and we've been living with them for so long. And so here's an example of a process of fermentation that we use. So this is a microscopic view of a yeast, Saccharomyces cerevisiae. And so yeast are a microorganism because you need a microscope to be able to see them, but they are not a bacteria. And so, we'll talk more about what are the characteristics of those different types of organisms coming up. But here's a yeast. So baker's yeast is Saccharomyces cerevisiae, an actual organism. When you buy baker's yeast, it looks like just a dry powder, but each of those little dots is an organism, is a yeast cell. And so when they get activated And maybe you might have seen people have sourdough starters, for example. That's basically like a culture of yeast that you are feeding and keeping going so that it can do fermentation for you and make the sourdough bread. So here is, an example of like a sourdough starter. And the yeast doing this process of fermentation make ethanol, but also carbon dioxide. So these little bubbles are bubbles of carbon dioxide. And that's what makes the bread like have those small, tiny little holes of air that it looks like to us, but that actually like is a pocket that comes from these bubbles of carbon dioxide that kind of like pop during the baking process. So we use yeast and let stuff like rise so that there's time for the carbon dioxide to make those bubbles and make something fluffier. So the yeast cells metabolize and break down the carbohydrates in the flour that you feed it, and then make the carbon dioxide that makes the bread rise and be like fluffy. So yeah, long before the invention of the microscope, some people theorized that infection and disease were spread by living things. A disease doesn't kind of just happen. Some people had an idea that, okay, maybe there's something behind this disease happening. So some people theorized that infection and disease were spread by living things that were too small to be seen. And some people also correctly intuited, like they figured out somehow. And so long before the invention of the microscope, before we saw that there were microorganisms, some people did theorize that infection and disease came from living things that were too small to be seen. They were able to figure out that it's not that diseases just happen, there's something behind them that kind of causes them to happen. And so some people also correctly intuited or like figured out certain principles regarding the spread of disease and immunity. So, there's something behind this disease and it can be passed on from person to person. And so in, records of history, there were methods of kind of quarantining people away so that they wouldn't be able to spread the disease to other people. Or that maybe once you have a disease, it's less likely for you to get the same disease. So some people, were onto something. Again, without knowing and seeing these microorganisms, they kind of figured out that something was behind these things and gets passed on to other people. And so the textbook is going to mention a lot of history of microbiology, a lot of different people, but I, in lecture, am going to highlight kind of some key people. So one of them being Antony van Leeuwenhoek. So he was the first guy to make observations of bacteria. So he made a very simple microscope, and it's kind of crazy that this is what it looks like right here in panel B. So Antony van Leeuwenhoek, using a microscope, was the first to actually see and describe observations of bacteria in 1975. At the time, he didn't know what they were. He just saw things in samples of water. And so he didn't know, he didn't have like a term for them yet. So he came up with the term animalcule. So it's like a molecule, but it's almost like an animal. And so now we can know, oh, that was probably bacteria. And then there's another guy, Robert Hooke. And so he kind of updated the microscope. So neither of these guys invented the microscope. It's kind of hard to tell who actually invented them because of history. But they get all the credit because they published things. And so for sure there are records of these guys. So anyway, Robert Hooke made some updates to the microscope. And so he made a compound microscope. So a simple microscope just has one lens. And we'll get back to this at the end of this lecture. And then a compound microscope has two lenses. And so Robert Hooke had a compound microscope, and he was the first to observe cells. And he was the one that named them cells, actually. So he was looking at a piece of plant, of cork, And so when he was looking at these observations, he saw these little tiny squares. And these squares he thought kind of looked like, not prison cells, but I think it was in the monastery, their rooms were called cells. And so that's why he called them that. Like it's like one little cell because it's one little like square room in a way. And so he named them cells. So he was kind of the first guy to describe cells, even though Van Leeuwenhoek was looking at bacteria, and bacteria are single-celled organisms. That's not kind of what he was paying attention to. So yeah, we say that he looked at bacteria, and then Robert Hooke did look at cells and name them, cells. And then during what is like referred to as the golden age of microbiology from 1857 to 1914, microbiologists discovered many new connections between the fields of microbiology and medicine. So these tiny little things that now we can observe and how they have effects in, human diseases and pathologies. And so another really important guy is Louis Pasteur. So he showed that there are different strains of microbes, and they each have different unique properties. So even though they're all tiny, he noticed, okay, yeah, they have differences, just like, bats are different than birds and different than other, like bugs. So even microbes have unique properties. He also noticed that fermentation is caused by microorganisms, and he invented pasteurization. So pasteurization is this process you might have heard about. If you look at like a milk gallon, you might have seen, pasteurized milk. So pasteurization is a form of microbial control using heat that is applied to foods. And so this process kills pathogens, and pathogens are microbes that make people sick. So it kills pathogens and reduces the number of spoilage-causing microbes while maintaining food quality. So this guy did us all a huge favor. And then one more microbiologist is Robert Koch. And so he was the first to show the connection between a single isolated microbe and a known human disease. So even though we had ideas of something's causing this disease, he was the one to be able to identify which microbe causes which disease for a few cases. So for example, he was the one that noticed that, okay, anthrax is actually caused by a bacteria, a microbe, Bacillus anthracis. So he found a few others as well, and he was able to pinpoint, the microbes that were causing a particular disease for a handful of diseases. And he came up with Koch's postulates, which is kind of the methodology we still use to be able to identify the same way. Okay, here's a disease. How do we identify what bacteria is behind it? Yeah. So that was kind of like the history of the field of microbiology, like what we knew and how we kind of came to know what we do. That was just a super overview. Again, the textbook has more information that is kind of interesting to get into and follow that path of history. So now we're going to talk about some stuff that we do know now. So microorganisms, again, they're just small organisms. And even though they're small, they vary in all sorts of ways. From their size, their appearance, and their rates and methods of reproduction. And so as we learn more and more about these organisms, we have to be able to classify them. We can't just say, oh, it's a microbe. There's so many now, and we know so many different things about different categories. So we need a way to systematically organize these organisms. So taxonomy is this field that involves the classification, description, identification, and naming of all living organisms. Living in the past. So being able to kind of categorize and organize all these different organisms. And so classification is the practice of organizing these organisms. into different groups based on their shared characteristics. So for example, us, like as mammals, we're mammals because we have hair and we have mammary glands and we give birth to live young instead of eggs. So based on things, we can like put them in a category, right? And so another famous scientist is this guy, Carolus Linnaeus. He was the most early taxonomist, so he was trying to figure out, okay, there's all these living organisms, how do we kind of like group them to organize them and study them efficiently? So he proposed this system that's now known as the Linnaean taxonomy. And this is a system of categorizing and naming organisms using a standard format so that around the world we can know what organism we're referring to. Even though we might have different casual names for things, if we have all around the world a standard naming system, then no matter where a scientist is, you can communicate and study the same organism and know you're talking about the same thing because there's a specific system. And so when he started, there was a certain understanding of organisms, and our understanding of organisms and categories have changed over time. So the system has been refined, but we still just kind of call it the Linnaean taxonomy. Even if it's not following the exact system he used back then, he gave a good scaffold for us to refine this system. So phylogeny is the evolutionary history of a group of organisms. So if you think about, again, mammals, because we're kind of, very human-centric. So if we think about mammals, the phylogeny would be kind of the evolutionary history of this group of organisms, the mammals. How did we branch off to different kinds of primates? So that's phylogeny. And so we can help show these evolutionary relationships using a diagram called a phylogenetic tree. And so scientists build these phylogenies and then show them through phylogenetic trees based on analyzing RNA, DNA, and proteins. And that really helps tell us about how organisms are related. Even if things look visually or not, if you analyze these molecules, it'll tell you a lot about how they're related or not, or how distantly. So scientists use these analyses of RNA, DNA, and proteins to help determine how organisms are related. The more similar these nucleic acids and proteins are between two organisms, the more closely they're related, they're considered to be. So here's an example of a phylogenetic tree. So this is definitely not all-inclusive. This is kind of just showing you the big picture. So this is it kind of looks like a tree, right? So this is a long distant common ancestor between these overall groups. Before they diverted. And so there was some kind of diversion and a group formed and kind of went this direction. And this group kind of went this direction. This group branched off and started this group. And so now we can, again, based on the comparison of DNA and RNA and proteins, lump them into these overall groups: bacteria, archaea, and eukarya. And then, so you first sort organisms into one of those three groups. And then within those groups, there are other groups. Okay, now it's sorted into eukarya. We can sort something into plants, animals, fungi, protists. So this is just an overview of the phylogenetic tree of life showing evolutionary histories. And so, as I mentioned, all organisms are grouped into those three major groups, and those groups are called domains. So domain archaea, domain bacteria, and domain eukarya. So let me go back for a second. So this purple domain, bacteria, archaea, and eukarya. So any organism that currently exists, and then any that we learn about, we first sort okay, what characteristics does it have? Does it go into this, this, or this? And then you can sort it even further. So within those domains, organisms are further subdivided into a hierarchy of specific like labels, levels, and sublevels. So these are the different categories. So the main category is domain. And then within that category, this huge one, domain, there are different kingdoms. So then you sort them into different kingdom. The next level is phylum, class, order, genus, and species. So these words are offset to kind of help show that this is inside of this group, is inside of this group, is inside of this group. So domain, kingdom, phylum, class, order, genus, and species. You find a new organism and you first, sort it into domain. Okay, within this domain, what characteristics does it have? So then what kingdom do we put it in? And then phylum and class, order, genus, species. And so on the next slide is an example. So say a dog. We are thinking about, let's think about a wolf. So a wolf, let's first sort it into one of the three domains. And so because its cells have nuclei, it would be in domain eukarya. And so here's a bunch of other stuff that's also within domain eukarya. So this is like a huge group. There's a lot of different things in here. The wolf, dog, lion, seal, whale, bat, fish, snake. A lot of stuff that's like, well, it's not that similar, but because they have nuclei in their cells, we put them in this huge group. The next is kingdom Animalia. So based on how this organism gets its energy, we can sort it into kingdom Animalia. It consumes other things, so it can't make its own energy. So there's some stuff that we kind of rule out. So this group gets a little bit smaller because less things fit those characteristics. Because it has a spinal cord, we can put it in phylum Chordata. So notice again, this group gets smaller and smaller. It's like ruling out other things that don't meet that. So it's kind of like 21 questions. And then, so that's how you sort it further and further. Class, Mammalia, order, Carnivora, family, Canidae, genus, Canis, and then species, Lupus. So the wolf has this specific name, Canis lupus. Because we've identified it and then gotten down to the genus and species, and that's what an organism's scientific name is the genus and the species. So it's always going to be two words for the scientific name, and that's telling you the genus and then the species. So that is binomial nomenclature. So bi as a prefix in words means two. Nomial means names. So the two names naming system. So a two-word naming system for identifying organisms by genus and species. And so for another example is us as humans. We're Homo sapiens. And so that kind of tells you that we're in the genus Homo, and there are other species within that group. And we're specifically Homo sapiens. That's our species. And so the binomial nomenclature or the scientific name is always going to be in italics. So that's why it's in italics here. And I always try to make sure that that's correct. If that's ever incorrect on a slide, that's a mistake. So italics always. And then the genus is always going to be capitalized, but the species is not. And that's just how it is. So Homo sapiens, only the first one is capitalized and not the species name. So if we go back to the Canis lupus, it's the same thing. So anytime you write about an organism, including bacteria, it's going to follow that convention. Italics, genus, and species. So again, this system is to help eliminate confusion. It assigns each organism a single, unique two-word name that's recognized by scientists all over the world. So even if you have like a casual name, for example, a specific bird, if you're a bird watcher, there can be different like casual names for things, but the species, scientific name is always going to be specific. And you can abbreviate this, which we will definitely do in microbiology just because it's faster, especially to write things shortened. So the abbreviated form of the binomial name is going to be the first initial of the genus. So whatever the genus is, you'll just put the first letter, a dot, so followed by a period, and then the full name of the species. So for example, let's go back to Homo sapiens. We could just put H period sapiens, and that's abbreviated. Escherichia coli is shortened as E. coli, and that's how we commonly know E. coli. It's the abbreviated form of its scientific name. And then within one species of microorganism, it's super interesting because they can be the same species, but there can be different subtypes called strains. So they're still the same species. They're not going to get a different name, but there are different subtypes, and so those are called strains. So there are different strains of E. coli. So specifically, it's E. coli strain O157H7 that is pathogenic, makes people sick. And the infection usually originates from contaminated water or food. But there are other strains of E. coli that are found naturally in our gut and help us digest food. But they're still technically that species, E. coli, just different strains. So if you ever see like, oh, E. coli strain, and then like numbers like this, that's kind of telling you which strain it is specifically. So microbes can be difficult to identify and classify. They don't have outward facing things that we can use to help us identify them. So for example, again, let's say bird watching. You can identify a bird based on what the feathers look like, what the region is, what the beak looks like, what the feet look like, the size of the wingspan, things like that. So without like easily observable characteristics like that, bacteria are so tiny and they don't have that much physical things to tell them apart. They can be difficult to identify and classify. So scientists have to kind of get creative and come up with ways to study their biochemical properties to help identify and differentiate and then classify microbes. And so that's what you're going to be learning a lot about in lab. So in lab, we're going to be doing different biochemical tests that can help identify the chemicals that are unique to certain species. So yeah, again, they don't have as many outward physical differences for us to tell, okay, this is E. coli versus this. You have to use a lot of different things to try to figure out, okay, what is this? And that's literally what we're going to be doing in lab. And you will get a unknown bacteria and trying to identify it based on, does it do this kind of metabolism? Does it have this kind of cell wall? Can it break down this thing? So you are getting the practice of being like a microbiologist doing those things in lab. And so there is a set of manuals called Berge's manuals that are the standard references. They have this just like huge charts of different characteristics of different microbes that we know right now, so that if you do have like an unknown, or say you are in a clinical setting and you need to understand, okay, somebody has this particular microbe, I'm doing these tests to figure out what it is, you can refer to them and they'll tell you, okay, if you do this, 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 and this comes out positive, this is positive, then it's this kind of bacteria. So that you can identify what it is. And so again, those are called Berge's manuals, and we'll get to know them more in lab.